Okay, so welcome back to this next video in which we are discussing endothelium-dependent vasodilatation. Okay, right, so we're currently in the process of discussing these pro-contraction influences on the vascular smooth muscle cells. So we have discussed the fact that all the time in normal vascular physiology, the vascular smooth muscle cells surrounding arterioles are receiving inputs that are telling them to contract, and also inputs that are telling them to relax, and these inputs balance each other out, and normal vascular diameter is maintained. Okay, and if you want to push the arterioles towards vasoconstriction or towards vasodilatation, all you need to do is upset that balance by either increasing the procontraction forces and causing vasoconstriction, or by increasing the relaxation influences and therefore pushing the arterioles towards vasodilatation. Okay, right. So we're currently in the process of discussing the pro-contraction influences, and we've just seen the fact that neurotransmitters can activate ligand-gated ion channels on the surface of the vascular smooth muscle cells, which can cause graded potentials within these vascular smooth muscle cells, which literally just means a depolarization without an action potential. Okay, and that depolarization can then activate voltage-gated calcium channels, which are going to allow calcium from the extracellular fluid to come into the cytoplasm, and that can lead to your global calcium rise, which can then trigger the contraction by binding to calmodulin and then activating the myosin light chain kinase, which will then phosphorylate the regulatory myosin light chains and initiate the sliding filament mechanism of muscle contraction. Okay, right. Now what we want to do is talk about the other way that neurotransmitters can activate contraction in these vascular smooth muscle cells. Okay, so this is through G-protein coupled receptors that are GQ/11 coupled. So let's just have a look at this pathway. Okay, so we will now again show the membrane of the vascular smooth muscle cell as a phospholipid bilayer, so that I can draw you a G-protein coupled receptor in this vascular smooth muscle cell membrane. Okay, so this is the extracellular fluid here above, and then this is the cytoplasm represented below. Okay, right. So G-protein coupled receptors then have this characteristic 7 membrane spanning alpha helix structure, like so. And there are absolutely loads of these things in humans. In humans alone, there are over 800 of the things. Okay, in the entire animal, well, in fact, in all the kingdoms of life, we have found, I think, over 30,000 different G-protein coupled receptors so far. So it's going to grow, obviously. So this is a massive, massive family of proteins. So this is some G-protein coupled receptor. Okay, now, G-protein coupled receptors activate G-proteins, heterotrimeric G-proteins, once they have been activated, and different G-protein coupled receptors activate different heterotrimeric G-proteins. The G-protein coupled receptors on the surface of the vascular smooth muscle cells, which are going to be pro-contraction when they're activated, are going to activate certain heterotrimeric G-proteins known as GQ/11 heterotrimeric G-proteins. So let me just explain what that means after I've drawn you a picture of one of these things. Okay, and I haven't got quite as much space as I budgeted for because I've written that cytoplasm there. Never mind, I'll put this like so. Okay, so heterotrimeric G-proteins then, I'll write that full word out, heterotrimeric. Okay, so the name gives you a lot of clues about the structure of these things. Hetero means different, okay, trimeric means three-membered structure. Okay, so a heterotrimeric G-protein is going to be a three-membered structure where all of the three members are different. Okay, and indeed they're made out of an alpha subunit here, a beta subunit here, and a gamma subunit here, 3 separate subunits all bound together. Now, the alpha subunit is the subunit after which the entire heterotrimeric G protein is named. Okay, we don't care about the beta and the gamma subunit. And I should probably say before that statement that there are absolutely loads of different types of G protein alpha subunits, G protein beta subunits, and G protein gamma subunits. Okay, so now I'll bring that statement in, that the naming of a heterotrimeric G protein is completely determined by which alpha subunit you use, not which beta or gamma subunit you use. And the reason for that is that the signaling properties of the heterotrimeric G protein seem to be completely determined by which alpha subunit you use and not which beta and gamma subunit you use. So there is a logic in just naming heterotrimeric G proteins according to which alpha subunit they have. So in a GQ/11 heterotrimeric G protein, the alpha subunit is going to be what's known as alpha Q/11. Now what does that actually mean? Well, that means that it could be alpha Q. Okay, alpha Q is a specific alpha subunit of a heterotrimeric G protein, or it could be alpha 11. Alpha 11 is another alpha subunit of a heterotrimeric G protein. Okay, a far less famous alpha subunit than alpha Q. Alpha Q is much more famous, but actually it's almost identical structure to alpha Q, and it does exactly the same thing. So this could be alpha Q or alpha 11. So that's what that means when people put alpha Q/11 like that. Okay, right. Now, initially the heterotrimeric G protein is going to be in the off state, and this is a running theme for all G proteins, not just the heterotrimeric ones. Okay, G proteins have two states. They have an on state and an off state. Now in the off state, which is the state that it's originally going to be in, the alpha subunit has GDP bound to it. Okay, so in the off state you have GDP bound, and then in the on state you instead have guanosine triphosphate GTP bound. Okay, so here is our heterotrimeric G protein. At the moment, the alpha subunit is in the off state and therefore has GDP bound. Okay, now when the alpha subunit is in the off state, it is capable of binding to the beta and the gamma subunit. Now, the beta and the gamma subunit always remain bound together, and that's the reason that they together are known as the beta-gamma complex. But the alpha subunit can dissociate from the beta-gamma complex, and it's only going to be bound to the beta-gamma complex when it is in the off state and has GDP bound. Okay, so I'll colour in the beta and the gamma subunits here. Okay, the other thing I want to say is how does the heterotrimeric G protein attach to the underside of the phospholipid bilayer? Well, both the alpha subunits of heterotrimeric G proteins and the gamma subunits of heterotrimeric G proteins actually have lipid moieties attached onto them. Okay, so these little lines that I'm showing here, these are lipid molecules that you have attached onto the protein. Okay, so one onto the alpha subunit and one onto the gamma subunit, and these have implanted into the inner leaflet of the phospholipid bilayer, and that's how the whole heterotrimeric G protein is attached to the underside of the phospholipid bilayer by these lipid moieties. Okay, right, we're not going to go into the specifics of which lipids are attached. The basic principle is that you've attached a lipid molecule on there. It's now very insoluble, so implants into the inner leaflet of the phospholipid bilayer, and that's how the heterotrimeric G protein is attached there. Okay, so I think I've done enough introduction then. Let's now talk about this pathway. So, if we've got some G protein coupled receptor here that does indeed couple through the GQ/11 heterotrimeric G protein, then when you stimulate this G protein coupled receptor with its ligand, okay, many neurotransmitters will work through these G protein coupled receptors which are coupled to GQ. So an example would be the neurotransmitter acetylcholine, ACH, okay, which has the G protein coupled receptor M3, which is GQ/11 coupled. Another example would be noradrenaline, NA, which has the alpha-1 adrenoreceptor, which is GQ coupled. Those are both GQ coupled G protein coupled receptors. Okay, which respond to neurotransmitters. Okay, once that neurotransmitter or ligand molecule more generally has bound to its G protein coupled receptor, what will happen is the G protein coupled receptor will change conformation, okay, and it will now be able to interact with the heterotrimeric G protein. It will now be able to bind to the heterotrimeric G protein, and what it's going to do is it's going to catalyze the turning on of the heterotrimeric G protein. So it's going to catalyze the dissociation of the guanosine diphosphate from the alpha-Q/11 subunit, and then what will happen is that guanosine diphosphate will be replaced by a guanosine triphosphate molecule. Okay, so it's the G protein coupled receptor that catalyzes this transfer of guanosine diphosphate for guanosine triphosphate. Okay, and when you bind guanosine triphosphate to the alpha-Q/11 subunit, it now goes from being in the off state to being in the on state. Now, when it's in the on state, it changes conformation. And this change in conformation means that it dissociates from the beta-gamma complex. Okay, so drawing this again, what's going to happen is the alpha-Q/11 subunit is now going to dissociate from the beta-gamma complex. Okay, it's also going to dissociate away from the G protein coupled receptor. And both the beta-gamma complex and the alpha-Q/11 subunits are now going to go their separate ways. So here is the beta-gamma complex going off, still attached to the underside of the phospholipid bilayer because of the lipid moiety that's attached onto the gamma subunit of the beta-gamma complex over here. And then the alpha-Q/11 subunit has also gone off on its own. Okay, now both of these subunits can actually have signaling roles, but we're specifically going to be interested in alpha-Q/11's signaling roles. Okay, so alpha-Q/11 is now in the on state with guanosine triphosphate bound to it. What does it now do? Okay, well, it's going to activate an enzyme known as a phospholipase C beta enzyme. So let me just write this here. So phospholipase C beta enzymes. Now there are a huge number of different phospholipase C enzymes. Phospholipase C beta enzymes is a family of phospholipase C enzymes which has 4 different members in: phospholipase C beta 1, phospholipase C beta 2, phospholipase C beta 3, phospholipase C beta 4. Okay, all coded for by separate genes, but they all have this same property that they're activated by alpha-Q/11 subunits in the on state. Other families of phospholipase C, such as the phospholipase C gamma enzymes and the phospholipase C zeta enzymes or eta enzymes or delta enzymes or epsilon enzymes, there's loads of the things. Okay, those are not activated by the alpha-Q/11 subunits. Okay, so this family, PLC beta enzymes, are the ones that are specifically activated by alpha-Q/11. And as I say, there's 4 different members of this family: 1, 2, 3, and 4. Okay, but we're not going to dwell on that point. For our purposes, it's just a phospholipase C beta enzyme. Okay, now, phospholipase C beta enzymes are all the time moving between being in the cytoplasm and being attached to the underside of the phospholipid bilayer. So if this is our phospholipase C beta enzyme here, sometimes they'll be hanging around in the cytoplasm, at other times they will have attached to the cell membrane here. Okay, however, they will not be active. They're never active. They're just moving between 2 different locations. Okay, the alpha-Q/11 GTP subunits can activate those phospholipase C beta enzymes that are currently attached to the underside of the phospholipid bilayer. Okay, so those phospholipase C beta enzymes that are currently in this location can be then acted upon by the alpha-Q/11 with the GTP bound to it. And what's going to happen is the alpha-Q/11 is going to activate the phospholipase C beta enzyme. Okay, right. The phospholipase C beta enzyme is then going to break down a membrane phospholipid, so something that is a component of the phospholipid bilayer. Okay, and this membrane phospholipid is for short called PIP2 or PIP2. Okay, but its full name is, get ready, phosphatidyl inositol. Okay, that's the P and the I, so the first P is for phosphatidyl. And don't worry, I will explain the structure and the name of this thing to you in a moment. So this is phosphatidyl, okay? The I is then for inositol here, okay? And then it's phosphatidyl inositol, 4,5, okay? And then it's bisphosphate. Okay, so the P2 on the end means phosphate, and then because you've got two phosphate groups, it's P2. Okay, so this P2 is for the bisphosphate, the 4,5 bisphosphate. Okay, now some people actually extend this name out to phosphatidyl inositol, and then they'll put brackets like this, 4,5 P2. Okay, now that's more rigorous, okay? That makes sure that people know what you mean here. However, if you just say PiP2, people will assume that you mean 4,5 bisphosphate. They will not assume that you mean 3,4 bisphosphate or something like that. Okay, they'll assume that the phosphate groups are on the 4th and the 5th carbons of the inositol ring. Okay, because this is the most famous PIP2, this is the main one. Okay, the other forms of PIP2, where the phosphates are of different carbons, are much more niche areas of knowledge. Okay, so if someone was talking about one of those, they would stress which carbons it was off by using this notation. But if someone doesn't stress which carbons the P2 phosphate groups are coming off, then you can assume that it is this form. Okay, right, so this is a membrane phospholipid. It's a normal component of the phospholipid bilayer. Okay, now it's quite a rare component of the phospholipid bilayer. There are much more common components of the phospholipid bilayer, one of which we'll see later on when we come to discuss the arachidonic acid generation. Okay, but it is a component of the phospholipid bilayer. So let me now show you its structure. Okay, and it is a phospholipid. Okay, remember the only characteristics you need in order to be a phospholipid are you need a phosphate group and a lipid group. Okay, and this certainly has a phosphate group and a lipid group within it. Okay, so let me draw you a little picture of this then. Okay, so I'll start, in fact I might actually show this in the membrane, so I'll draw a picture of the membrane here, and now we'll draw it as one of the parts of the inner leaflet of the phospholipid bilayer. Okay, so I'm going to start off with this phosphatidyl portion of the name. Okay, now what that means is that you have got a structure known as phosphatidic acid, okay, as your basis for building this larger molecule. Okay, so PiP2 contains a molecule known as phosphatidic, I was about to miss this bit off, phosphatidic acid there. Okay, so this is my little schematic picture of phosphatidic acid, my little cartoon of it. Okay, this horizontal line that is now coloured in green here, this is going to represent a glycerol molecule. Okay, now remember the proper chemist's name for glycerol is propane 1,2,3-triol. Okay, and although that's much more of a mouthful than glycerol, it's a useful name because it tells us exactly what this molecule is. It's a 3-carbon molecule with 3 alcohol groups coming off it. Okay, right, now these 2 vertical lines that are now coloured in orange, these are going to represent fatty acids, okay, which remember are long-chain carboxylic acids. So fatty acids is the sort of colloquial name, whereas the more official name is long-chain carboxylic acids. And again, long-chain carboxylic acids is a more useful name because it tells us exactly what these things are. They're carboxylic acid molecules which have a really long tail, okay, and it's generally very hydrophobic, and this is the lipid groups of this phospholipid molecule, and this is the reason it's anchored in the phospholipid bilayer in the first place. Okay, now these long-chain carboxylic acids have been esterified onto the first and the second alcohol groups of the glycerol molecule by normal ester links. Okay, then off the final alcohol group that comes off the third carbon of the glycerol molecule, you have attached this little blob in purple, which represents a phosphate group. Okay, so this is the phosphate group of the phospholipid. So even now we've already got a phospholipid structure. Okay, phosphatidic acid on its own is a phospholipid. Okay, so this is a phosphate group here. Okay, and it's been attached to that alcohol group by a phosphoester link. Okay, so let me just make sure that people are aware of what a phosphoester link is. So let me show you the structure of phosphate group firstly. So a phosphate molecule looks like so. Okay, you have a phosphorus atom at the centre with an oxygen double bound to it coming off here, 2 alcohol groups generally coming off either sides, and then an oxygen coming off the phosphate group that has acquired an electron via ionic means and therefore has a negative charge. In effect, you can view this as another alcohol group that has just lost its proton. Okay. Now, what you'll notice is if we look at, for instance, this group here, this looks extremely like a carboxylic acid group. In fact, if that phosphorus atom was replaced by a carbon atom, that would be a carboxylic acid group. And in fact, this can react with alcohol groups in pretty much the same way as carboxylic acid groups can, okay, to form something very similar to an ester link, except that it's now called a phosphoester link. Okay, so that's how you've attached that phosphate group onto that alcohol group of the 3rd carbon of the glycerol molecule. Okay, right, so that's phosphatidic acid. Now that's only the first part of phosphatidyl inositol 4,5-bisphosphate. That's the origin of the phosphatidyl right at the start of the name here. What we now need is this inositol portion of the molecule. So what you're going to do is you're going to attach onto this phosphate group here an inositol molecule. Okay, now an inositol is a 6-carbon molecule. Okay, so let me tell you what the actual official name for inositol is. So inositol is the old biochemist name for a molecule that would now be called cyclohexane 1,2,3,4,5,6 Okay, and that's certainly a much bigger mouthful than inositol, but it's a more useful name because it again tells us exactly what this molecule actually is. It's a 6-membered carbon ring, which is why I've drawn a hexagon here. Okay, this is representing the 6-membered carbon ring, and off each one of the carbons of the 6-membered carbon ring, you have an alcohol group. Okay, so if we draw the skeletal structure of it here, and remember in skeletal structures you don't show carbon atoms, they're implicitly shown by corners, and you don't show hydrogen atoms coming off carbon atoms. Okay, so here is our 6-membered carbon ring. This is cyclohexane. All of the bonds between the carbons are single bonds. Okay, and then off every single one of these carbons you're going to have an alcohol group and also a hydrogen atom coming off. Okay, now this is more difficult than it might seem. You might be thinking, oh, how wonderful, what a simple molecule. But there's a subtlety here. There is a subtlety here, unfortunately. And the subtlety is optical isomerism. Okay, and we're actually going to need to go into this to understand why PIP2 is called phosphatidyl inositol 4,5-bisphosphate. To understand that 4,5 in the name, we need to understand the optical isomerism of this molecule. Okay, so let me try and make you appreciate the problem here. Okay, if we imagine that this 6-membered carbon ring here is perfectly sitting within the plane of the piece of paper, so all of those 6 carbons in this beautiful hexagon are perfectly in the plane of the piece of paper, then the other 2 bonds that come off each of those carbons are going to be coming off out of the page and into the page respectively. So one bond is going to be, if we look at this carbon here specifically, one bond is going to be coming off at an angle like that my pen is currently making, and the other is going to be going into the page at an angle like this. Okay, so then the difficulty becomes, so do you have the alcohol group coming out of the page towards us and then the hydrogen going in to the page away from us, or do you have it the other way around? Do you have the hydrogen coming out of the page towards us and then the alcohol group going into the page away from us? And then you have to factor that in for all 6 of these, and then suddenly it becomes a much bigger nightmare. There are loads of different forms of this molecule that are all optical isomers of one another. Now, luckily, there is only actually one optical isomer of inositol that is used in biology, whippy, and this is what's known as myo-inositol. Okay, now generally if you're talking to a biologist, they will not call inositol, well, they will not call myo-inositol. They will just refer to it as inositol, and they will assume that you realise that they are talking about the only optical isomer that's actually used in life, which is myo-inositol. Okay, it's only when you talk to a chemist that they actually say specifically myo-inositol, because they actually work with some of the other isomers of inositol. Okay, right, so let me show you then the structure of myo-inositol by showing you the orientations of the alcohol groups coming off each of these carbon atoms of the cyclohexane ring. So, this one here, we'll start off with this one here. The alcohol group is going to be coming out of the page towards us off this one here. And the way I show bonds coming out of the page towards us is that the bond gets thicker towards the end. Okay, so this is an alcohol group coming out of the page towards us. Then off this one here, again the alcohol group is going to be coming out of the page towards us. Off this one here, again it's going to be coming out of the page towards us. Okay, so it's all going simply so far, but now we're going to switch. Now the alcohol group off this one here is going to be going into the page away from us. Now note that I am not showing the hydrogen atoms coming off each of these carbons because I said we're drawing a skeletal structure. So all I need to show you is where the alcohol groups go, and then you can work out that the hydrogen groups go the other way, basically. Okay, so this one, the alcohol group goes into the page away from us. This one here, the alcohol group comes out of the page towards us, and then finally this one here, the alcohol group once again goes into the page away from us. So these two are oppositely orientated to these four basically, so this is it's kind of nicely symmetric down there. Okay, now this carbon here is known as carbon number 1, this carbon here is known as carbon number 2, this carbon here number 3, this one here 4, this one here 5, and this one here 6. That is the numbering system for the carbons of myo-inositol, set by chemists, I presume. Okay, so this inositol molecule here is going to be a myo-inositol molecule. So now the question is, which carbon of the myo-inositol ring have we attached onto this phosphate group via a phosphoester link? And I should probably just mention that even once one of these alcohol groups has been used up in a phosphoester link, you will still have another one here. So phosphate groups are almost like two-headed carboxylic acid groups because they can actually form two phosphoester links. And in fact, this phosphate group is forming another phosphoester link with the alcohol group of the first carbon of the myo-inositol ring. So it's the first carbon here. Okay, now that's then what we have created so far is phosphatidylinositol. It's phosphatidic acid linked to inositol. But we want phosphatidylinositol 4,5-bisphosphate. So what we want to do is add 2 more phosphate groups onto this molecule to create PIP2. And which carbons are we going to add them onto? Well, it's the 4th and the 5th carbons. So you're going to stick a phosphate group coming off this 4th carbon's alcohol group, and you're also going to stick a phosphate group coming off the 5th carbon's alcohol group, like so. Okay, so the reason that I went into this discussion of the optical isomerism of myo-inositol was so that you can understand why this molecule is not phosphatidylinositol 3,4-bisphosphate, because if I had just drawn this cartoon, you would probably say, well, why is that not called the 3rd carbon? Well, it's specifically the 5th carbon that the phosphate group comes off rather than the 3rd carbon. And there is a difference between the 5th carbon and the 3rd carbon, which I hope you can see. These two are not symmetric down that line. Okay, this one is different from this one. Okay, the 5th carbon has 2 carbons with alcohol groups going off in opposite directions to the direction which its alcohol group comes off. The 3rd carbon doesn't. It has a neighbouring carbon which has its alcohol group coming off in the same orientation as it. So there is that subtle difference between the 3rd and the 5th carbon, and in PIP2, phosphatidylinositol 4,5-bisphosphate, the phosphate group comes off the 5th carbon, not the 3rd carbon. Okay, right, so that was a bit of a laboured explanation for that. Right, so that's phosphatidylinositol 4,5-bisphosphate. Back to phospholipase C-beta then. Phospholipase C-beta is going to break down this molecule, and going through this pathway is extremely important because this underlies a lot in this whole system. Okay, we're going to see this occurring in endothelial cells as well, and we're not obviously going to go through the entire pathway again, but we'll just say when we do it in endothelial cells, all of this happens. Okay, so it is important. Right, so phospholipase C-beta is going to break down phosphatidylinositol 4,5-bisphosphate in the phospholipid bilayer. And as I say, it is a normal component of the phospholipid bilayer. Cells do have a little bit of PIP2 in their leaflets. Okay, right. So what's going to happen is when phospholipase C-beta is activated by the alpha-Q/11 subunit in the on state, it's going to break down the PIP2 molecules. Specifically, it's going to cleave this bond here between the third alcohol group of the glycerol molecule and the phosphate group down here. So it's going to cleave that phosphester link, and it's therefore going to create two separate products. So let's have a look at these two separate products. In fact, I think I'll go onto a new piece of paper for this. Okay, right. So we'll need our old piece of paper so that we can see. Oh, actually, I'll just draw out the molecule again. Right. So let's just redraw out our phosphatidylinositol 4,5-bisphosphate quickly. So here are our two long-chain carboxylic acids, the glycerol, the phosphate group, the inositol ring here, and then the two phosphate groups coming off the fourth and the fifth carbons. Okay, so let's colour this in again. So here are the long-chain carboxylic acids. Here is the glycerol molecule in green. Here are the phosphate groups in purple. And here is the inositol ring in blue here. Right, so phospholipase C-beta then is going to cleave PIP2 across there, okay, and break that phosphoester link. And the two products of this cleavage are going to be a glycerol molecule with two long-chain carboxylic acids attached to it. So here is the glycerol molecule, and here are the two long-chain carboxylic acids attached there. And this product is known as diacyl glycerol. Okay, so diacyl glycerol, and for short, it's abbreviated down to DAG. Okay, now the reason it's called that is because when you esterify a long-chain carboxylic acid onto any old alcohol group, you actually only attach on the acyl group of the carboxylic acid. So let me just remind you what an acyl group is. Okay, so if this is the carboxylic acid here, and I'll just put an arbitrary R group here, if we have the alcohol group of the glycerol molecule here, okay, so this is our alcohol group of the glycerol molecule, this is our R group of the long-chain carboxylic acid, now colour-coded. When you esterify the long-chain carboxylic acid onto the alcohol group, what you actually do is you take the alcohol group off the long-chain carboxylic acid, you take the hydrogen off the alcohol group of the glycerol molecule, and then you attach the carbon of the carboxylic acid group onto the oxygen of the alcohol group, and this is the ester link that we formed here. Okay, so you haven't actually added on the entire carboxylic acid molecule. Instead, what you have added on is this portion here, which I've now outlined in blue. This is what's referred to as the acyl group of the long-chain carboxylic acid, or in fact of any carboxylic acid. Okay, so that's why this is called diacyl glycerol, because you've attached two acyl groups onto glycerol, and that's the origin of that name. Okay, right. The other product that you then produce is IP3, inositol with with three phosphate groups coming off it. Okay, so here it is. Sorry about that little blotch there, that sort of made it look as though I'd put a double bond there. I'll fix it by colouring it in. Okay, so here is the inositol ring in blue, and now you've got three phosphate groups coming off the first, the fourth, and the fifth carbons. Okay, so this is called inositol, and some people will call it myo-inositol, inositol 1,4,5-trisphosphate, and as I say, some people would call this myo-inositol 1,4,5-trisphosphate if they were being extra rigorous. Okay, but this is usually just abbreviated down to I for inositol, P for phosphate, and then you put a 3 there for 3 phosphate groups. Okay, right. Now, both of these two products of this cleavage actually have important signalling roles. Okay, however, the only one that we're going to care about is IP3, inositol 1,4,5 trisphosphate. Okay, this is going to drop into the cytoplasm whilst the diacylglycerol is going to remain in the inner leaflet of the phospholipid bilayer. Okay, now what is the IP3 then going to do? Well, it's going to activate IP3 receptors in the membrane of the sarcoplasmic reticulum. Okay, so if I draw the entire smooth muscle cell again, okay, a key intracellular organelle within the smooth muscle cells is the sarcoplasmic reticulum here. So this is representing the SR, which stands for the sarcoplasmic reticulum. Now I'll just move this up. Now the sarcoplasmic reticulum is the equivalent of the endoplasmic reticulum in a non-muscle cell, but in muscle cells it's got this special name, it's called the sarcoplasmic reticulum. Now, you remember I told you earlier that the cytoplasmic calcium concentration is very low, around 100 nanomolar. Well, the calcium concentration is much, much higher than that in the lumen of the sarcoplasmic reticulum. Okay, so you have this intracellular store of calcium ions in the sarcoplasmic reticulum lumen. Okay, now, there are special channels which can allow calcium to leave the lumen of the sarcoplasmic reticulum and return back into the cytoplasm. Okay, now one of these such channels is what's known as an IP3 receptor. Okay, which I'll just abbreviate down to the IP3R for IP3 receptor. Okay, and this is activated to open by IP3. It's a little bit more complicated than that, but roughly, as a decent approximation, IP3 leads to the opening of the IP3 receptor. As I say, the actual gating mechanism of IP3 receptors is still not known, okay, but we know it's more complicated than just IP3 binding. Okay, so IP3 is going to drop into the cytoplasm and it's going to act on IP3 receptors and it's going to lead to them opening. Okay, and that's going to lead to the release of calcium into the cytoplasm, which is then going to contribute to a global calcium rise, and then you're going to initiate contraction by calcium binding to calmodulin. The calcium-calmodulin complex is activating the myosin light chain kinase, and the myosin light chain kinase then phosphorylating the regulatory myosin light chain, leading to the initiation of the contraction of the smooth muscle cell. So this is the other way that you can activate the contraction of these smooth muscle cells, by producing a global calcium signal, and this global calcium signal has been produced in a very different way. Okay, rather than calcium coming from the extracellular fluid, you have liberated calcium from the intracellular stores. Okay, so that now concludes our discussion of the pro-contraction mechanisms. In the next video, we will actually finally get onto the topic of this video, which is the endothelium-dependent relaxation, okay, which leads to the vasodilatation. Okay, so we'll start off with a little discussion of what's going to be happening in the endothelial cells. Then we'll start by looking at the generation of nitric oxides. Remember, we discussed long ago how there were these three contributing mechanisms here. Okay, did I put them here? Yes. There were the three contributing mechanisms of endothelium-dependent relaxation: nitric oxide, prostacyclin, and endothelium-dependent hyperpolarization. We are going to start off with nitric oxide, which is the most important one. Then we will go on to prostacyclin, and finally we'll go on to endothelium-dependent hyperpolarization.